Before I start, I just want to make one comment with respect to that last observation. If you go from 5 to 10 fish to 300 fish or 3,000 fish, you still have an effective size that is based upon the 5 or 10 fish, and you still have a problem even if it doesn't manifest in the first generation. Significant one. I'd like to thank everyone for who organized this, for the opportunity to come here and tell you about the work we've been doing on genetic population structure of coho salmon over the last decade or so. I'm going to start at a very broad scale and kind of drill down to a very local scale to try to give people the context as we move into these very important discussions about what's going on in the Klamath Basin and in particular in the Shasta River. First off, I want to say that this work that I'm going to be talking about today is the product of a lot of effort by a lot of different people over a number of years in my research group and a lot of collaborators who provide us with samples. Okay, great. And I also want to say that our research group is a cooperative research group of the National Marine Fisheries Service, Southwest Fisheries Science Center, and the University of California, Santa Cruz. And we have somewhat equal numbers of people from those two institutions who work in the lab. So I'm going to start off telling you about the big picture. This is a diagram of population structure for the species as a whole, ranging from Russia all the way down to San Vicente in San Vicente Creek in Santa Cruz County, just right near our laboratory, which is the southernmost persistent population of coho salmon in the world. I should say in the native range. We've got them in Chile as well. And this is a massive amount of data, over 6,000 fish with 18 microsatellite genes and populations. A lot of these designations such as Alaska or BC or Puget Sound or the Columbia are conglomerates of a number of different populations. And there are a couple of things that I want to point out here. One is that well, I should say this is also, the California portion of the range is way overrepresented here because of where we sit geographically, not surprisingly. And the thing that I want to point out is that, and I should actually have an ESU map here, because what one would see if one knew how to look at these trees, which is really very difficult even for a lot of geneticists, is that the structure here corresponds very well with the ESU structure, the ESU boundaries that we use for management. And one thing I want to point out about these trees is that the important things about these trees are the branching patterns, the pattern of branching in these internal parts of the tree and the lengths of those internal branches. The lengths of the terminal branches are really not that important. Those have to do with sample sizes, sampling effects, and a variety of other things. So there's an important point to make here, which I'm actually going to make when I go to the next, actually, no, I guess I'll make it here then. And that is that the Klamath Trinity populations of coho salmon cluster most closely with the Rogue River and then with Wilson Creek, which is, as you know, sort of right outside of the Klamath River Basin, and that they are nested in the Sonk group, which you'll see a lot more decisively in the next few slides. And there is no indication of out-of-basin founding or ancestry of that stock. So this is just a little bit of an overview of coho salmon in California. We have two ESUs of coho salmon in California, as I'm sure you all know. They're both protected under the U.S. Endangered Species Act. The Southern Oregon, Northern California Coast ESU is threatened, and it includes populations from the Rogue down to the Eel River, and then the Central California, and actually a little, the Eel River, And then the Central California Coast ESU, which is ESA listed as endangered, stretches from there down to Santa Cruz County. They are, as you all know, and have been in decline for decades now and have been extirpated from a number of places where they previously occurred. And there are, in the places where they do occur, there are brood years that are either extinct or functionally extinct. And as you all know, the brood year variation in Coho salmon is very pronounced because most individuals spawn as 3-year-olds. So this is, oh, I'm sorry, this is actually not 6,600 fish. This is actually about 2,500 fish. And this is again drilling down to the level of California. And this is the same sort of a dendrogram that is summarizing relationships between populations of coho salmon in California, all the way from Wilson Creek, which is right on the California border, or near the California border, to down to Santa Cruz, right here. And a couple of things to note here are that all of the Klamath Basin, Klamath Trinity Basin samples all cluster together closely. It's a monophyletic group, and they are actually quite distinct from other fish in California. This is just another way of summarizing the same data that does away with the variation in length of the terminal, the end branches, and allows one to focus on the internal structure of the tree. And it uses a statistical resampling method to evaluate how much support there is in the data for those internal branches. And a few things to note. One is there's a very strong division between all of the populations north and south of what is the boundary of the Central California coast and the SONC ESUs, which is nice. And then again, within basins, local populations are all really quite closely related. And this is just this is a graphical representation in 3-dimensional space, sort of made 2-dimensional here, of individual genotypes from all of the fish in that previous slide. And I failed to point out, but actually all of these populations were sampled in the same year in the same cohort. So we've removed temporal variation in this study and just focused on the geographic variation. And again, what one sees is what I just mentioned. The primary axis of variation separates the CCC, the more southern ESU, from the SONC. And then within the SONC, there's a fair amount of differentiation between the Klamath stocks and the others, further to the south and even to the north. And again, this is reflected in this table here. And what this table is, it's the result of genetic stock identification, if you will, or what we have called assignment tests in the past. And what this is the evaluation of the genotype of an individual fish, one by one, in a maximum likelihood calculation, asking the question, what is the most likely population of origin of each individual fish? And if the answer is that it's the population in which it was sampled, then you get a tick right here along the diagonal, which is, for example, in the case of the Mattole River, indicates that a fish sampled in the Mattole is most likely to have originated in the Mattole. Or conversely, in this case, most likely to have originated in the Eel River for those two fish. And what I want to point out here is that, again, there's quite a bit of structure by basin in California. But when you look within basins, there's actually quite a bit of cross-assignment between different sub-basins or tributaries and local populations, indicating that there's a fair amount of gene flow between them. One way that one can interpret this kind of data is that these individuals that fall off of the diagonal are migrants, or the progeny of migrants. It's a little more complicated than that, but the values that fall off of the diagonal are indicative of recent gene flow between these populations. So again, you see that within basins, there's substantially more than between basins, for the most part. Okay, so now we're going to start drilling down to the Klamath River, and I was looking for good maps to use for this, and I came up with a couple. I decided to put this one up. I don't really need to tell you guys this, you kind of all know this. But I put this up for two reasons. One is because I want to stress that two things, that the basin is highly diverse, and a lot of the Coho salmon spawning habitat, in particular that we're talking about here, is on private land. Moreover, we forget now, and in fact the other map that I'm going to show you has the Klamath Basin sort of ending right here. Whereas we know that historically all of this was available to anadromous fish. I know not all of it because of barriers, but quite a bit. And in fact, ultimately, part of what we're dealing with now is undoubtedly due to the fact that we've restricted the amount of available habitat to Coho salmon to the bits that are now below these barriers to anadromy. Okay, so now we're going to get to the study area, which is those parts where fish can still display an anadromous life history, and talk about the sampling sites for what I'm going to be talking about next, which I think is what is of most interest to people, structure within the Klamath Trinity Basin. And I've indicated here the hot zone, as people call it, a very high disease prevalence part of the main stem Klamath between the Scott and the Shasta River. And the other thing I'll say is that these dots are rough locations in a lot of cases, particularly in the Scott River. We have samples from a variety of different tributaries, and to spare you all a sea of little dots, I've just put one dot for each of the tributaries here, McGarvey and Blue Creek and Sharber and Dutch and so on and so forth. So last year, actually I guess it was the year before last now, in 2010, we did some analyses of fish from the Klamath River Basin, including populations, or I should say broodstock from Iron Gate Hatchery, and we looked at all of these fish with 16 microsatellite loci and a very large number of fish, and provided some preliminary results. And those results were a little bit contentious, some of them, and were ultimately, I think, the basis for this conversation that we're having now. And so I'm going to actually start off by walking through those preliminary results, and then I'm going to talk about what we've done since then, which is primarily collecting more data on the same sets of populations. So what you see here, where you see IGH and a number, that refers to fish that entered Iron Gate Hatchery in that year. So these fish entered in 06, and these entered in 09, and these entered in 05, and 02. So these are adult fish, whereas a lot of the others are juveniles that were spawned by fish that returned in the previous year. And the thing that jumped out at us immediately about these results is the fact that you don't see strong signals of structure that are geographically consistent, but in fact temporal variation is the primary pattern that one sees in these data. This is again, try to focus on these internal branches because that's what's important in this context. This is the same analysis, but with data from a couple of different populations in both the the SONC and the CCC to sort of give us context and to root this tree, if you will. And again, just very strong signal of structure by temporal, by the year or the brood cycle, and not by geography. So remember how I showed the three-dimensional representation of all the individual genotypes from across California? Well, this is what you see when you look at the Klamath alone. Just a big muck of stuff. And in fact, what you see up here, those two, the blue and the green, which you may be able to distinguish from all these other dots, are the Trinity River populations. And this is all, everything from the Klamath right here, a big ball of undifferentiated stuff, genotypes. And in fact, the FST is probably the most commonly used measure of genetic divergence, genetic differentiation, and one can calculate it for each pair of populations or samples that one has. And it's numerical with 0 or slightly negative, indicating no differentiation at all, and higher values indicating more differentiation. And one needs to be quite careful about assuming too much or deriving too much inference from individual FST values for a variety of reasons I won't go into. But the patterns of different FST values from different pairs of different types of populations can be informative. And what we're looking at here is a frequency histogram of all of the pairwise FST values for different comparisons, types of comparisons of populations. Bogus Creek to an Iron Gate hatchery broodstock. Those are the yellowish bars. Iron Gate broodstock in 1 year to Iron Gate broodstock in another year. Those are the red bars. All of the Klamath River populations to Trinity River populations, blue, and green is the Shasta to Upper River, Upper Klamath meaning everything other than McGarvey and Blue Creek, to Shasta, or Shasta to everything else in the Klamath. And what I want to point out here is that within the Klamath, the highest FST values that we saw were between broodstock years at Iron Gate Hatchery. And in fact, the Shasta to other Klamath population values are kind of right there in the middle of everything else. In the interim period, we have done two things. We have substantially increased sample sizes by almost 1,000 fish, including some fish that were sampled, I guess, at Big Springs in the Shasta, some additional Scott River samples, and a few more years of broodstock from Iron Gate Hatchery. And in this analysis, as outgroups, we've replaced the Eel River with Ryan Creek, which is a little bit closer to the Klamath Basin than the Eel River. Oh, it's a Humboldt Bay trip. And what I want to point out is, in this tree is, again, don't pay attention to these long branches. It turns out that the Scott River samples are essentially homogeneous, and they'll be collapsed and some further graphics. But again, we just see this incredibly strong signal of structure by brood year, essentially. And I'll say more about that in a minute. Again, this is one of these statistical representations of the internal branches of these trees that help us to focus on how much data. Oh, I forgot one very important thing. So we added 1,000 fish, but the other thing that we did is we added almost 100 new genes to the data set for every single one of these fish. These are the SNPs that a gentleman from the Karuk tribe was mentioning. We've developed a whole new panel of SNPs for Coho salmon, and this is really the sort of first major application of those. And so we've got a huge amount of additional data relative to the preliminary results that we released last year. And again, we see essentially the same patterns. Everything is structured temporally and not by geography. So, oh, what happened there? I'm not sure. Well, there was something interesting there, but I don't know what it was. It looked okay on my screen. Let's go back for a second. Oh, I don't know what it is. In any case, so what I've done here is just in the interest of again focusing on what's going on in the Klamath River, I've removed the outgroups so that we can focus more on just the Klamath Basin populations and look to see what's going on. And in fact, again, we just see this incredibly strong set of relationships between individuals from the same brood cycles. And that totally makes sense, of course, because in 2010, the fish that are returning were born to the parents that were spawned at Iron Gate Hatchery in 2007, and those fish were born from parents that were spawned in 2004, and so on and so forth. So it makes sense, and in fact, we have used these SNP data to actually trace back and construct pedigrees between these individuals. I'm not going to be able to talk about that today, but it's actually incredibly interesting work. So again, I keep mentioning that the patterns that we see of structure are temporal and not geographic primarily. And it's been hard to tell whether that's true with respect to the natural populations, because I haven't actually told you when they were sampled or what brood cycle they correspond to. But in fact, when I map that onto this tree, what you see is that in fact, again, Bogus Creek the samples come from the 07-010 brood year. Not surprisingly, it clusters very strongly here. Scott River, same thing. The Shasta River is a mix of two different brood cycles, and not surprisingly, it sort of falls in between the two. Beaver Creek, sampled in 02, and that's the brood cycle that corresponds to this cluster here. And these, again, you see that the Trinity River populations are doing their own thing, and that the lower basin populations are actually more similar to them, really, than they are to the upper Klamath. So there are so many ways we can look at these data, and because this is such an important issue, we're going to look at them in a number of different ways, including the tree-based method that I just showed you, but also using model-based clustering. And what this technique does is it takes the genotypes of individual fish without any information about where they came from, where they were sampled, what subbasin, or anything, and it asks the question again in a likelihood framework: what is the most likely? What's the most likely affiliation of each of the individual fish, given a hypothesis about how many populations there are? In this case, the hypothesis is that there are two populations. K is shorthand for a population, and you allow individuals to be assigned fractionally to these different populations. These they're really not populations; they're clusters, but they often function as they often are coincident with population boundaries. And again, what I want to point out here is that the primary axis of variation here is between these different brood cycles. And to spare you having to go through a lot of these different colorful graphs, I'm going to jump right down to k equals 6. And again, the purple corresponds to one brood cycle at Iron Gate. The green corresponds to another brood cycle at Iron Gate. The yellow corresponds to another. And one thing I want to point out is that we've been talking about the fact that by and large jacks are not spawned at Iron Gate Hatchery, or they were not prior to 2010. Well, in 2011 and to a certain extent in 2010, they were spawned, and you can see them. They jump right out at you right there. And in fact, those individuals are jacks. So they come from this brood cycle, the yellow brood cycle. I'd also like to draw your attention to the Shasta River here. And what you see in the Shasta is highly mixed ancestry. Very, very, and you see that here as well. Again, the Shasta is a mix of a bunch of different stuff, and that's not surprising given what we know about straying. So this is another one of those the genetic stock ID assignment test tables, and I'm just putting circles here around the boxes that that set off all the Scott River samples and all the Iron Gate samples, so you can see that again there's just a tremendous amount of similarity between them, and this is and I should say this is when you don't actually ask the question, how likely is that assignment to the population of origin, but just what's the most likely one. But if we actually want to use a statistical, a sort of pseudo-statistical criterion for determining whether an assignment is high probability or not, then we can use this 90% probability criterion. And then you see that a lot of these, a lot of the assignments between different subbasins, particularly between the Trinity and the others, goes away. But there's still quite a bit of gene flow between Iron Gate and the Scott River and the Shasta River. And in fact, there actually are a few, let's see where there are, a few Trinity River fish that have been spawning in, well, at least one, in the Iron Gate broodstock. Actually, sorry, I looked at that the wrong way. These are these are Trinity River fish that have been spawned at Iron Gate over the years. FST, this metric that we talked about earlier for measuring genetic differentiation. This can be thought of roughly as a percentage of variation that is partitioned between pairs of populations. I realize you probably can't see these numbers very well, but for example, that number right there, 0.0002, means that essentially none of the variation is partitioned between the two populations, in this case two broodstock years, the same brood cycle of fish, and here it's more like 5% between, well, that's a bad comparison, between McGarvey Creek, I'm still on these out of basin ones, okay, and here we got about 1% between Iron Gate and McGarvey Creek. So not very much variation, and I've just highlighted here in red the ones between fish from different, from the same brood cycle at Iron Gate, and what you can see is they're all nearly zero. Again, very, very little differentiation between the brood cycles. I'm sorry, within brood cycles, and almost all of it partitioned between brood cycles. So again, we can plot the frequency distributions of these FST values in a frequency histogram and look at the relative frequencies. And again, we see that within the Klamath River Basin, the highest FST values are between broodstock from different years at Iron Gate. And that by and large, the differentiation between samples taken from different tributaries and even different years in the Klamath River Basin are very, very low. 1% approximately of the variation found in these populations is partitioned between it as opposed to found within it, which is like human populations, more or less. So now, so hopefully at this point I've convinced you that there's been a fairly strong influence of hatchery practices at Iron Gate, both on the Iron Gate stock, but also on natural populations in the Klamath River Basin. And so now I'm going to talk a little bit about the actual Iron Gate stock, and there is, I think, legitimate concern about what that stock, what that stock's effects on natural populations have been and are likely to be, and what the status of that stock is, whether or not it's got problems such that the effects that we see, the demographic effects, the genetic effects, are likely to be deleterious. And this really gets at some of the questions that were asked quite directly earlier about, you know, how can you tell if a stock is inbred? So what this represents is, in 2010, when we did this analysis, the last 3 years for which we had data on Iron Gate broodstock were 2006, 7, and 8. And what we did here is to calculate, based on the genetic data, the distribution, that's the bell curve-ish looking thing here, of relatedness values between all pairs of individuals that were returning to Iron Gate and used as broodstock. And what we did is we then said, okay, well, oh, and I should point out that for those of you not familiar with this, that pairwise relatedness is a calculation of what percentage of your genes you share with the other individual in that pairwise calculation. Full siblings share half of their genes, or they have a 50% probability of sharing any gene from identical by descent, and a relatedness coefficient of 0.5 is indicative of full siblings. 0.25 is indicative of half siblings. And we have sort of arbitrarily chosen the half sibling and above relatedness category as our hot zone, where we think these sorts of matings are going to create inbred individuals that are going to have a substantially lower probability of survival. And in fact, there's quite a bit of data about that I'll talk about tomorrow. And so what that means is that, in fact, in 06, 11% of the matings were expected to be, if in fact these individuals were randomly paired together, which they may not have been, 11% of the matings were expected to have been highly inbred. In 07, about 6.5%, and 08, about 8%. And keep in mind, this is if individuals that are returning and being spawned on dates that are completely random with respect to their family relationships. As I think Morgan mentioned, we suspect, we don't actually know. We have demonstrated this in steelhead, but we don't know. We will know soon. But we suspect that individuals from the same family are much more likely to return about the same date than any two random dates within the period of spawning for coho salmon, in which case this probability, this fraction of inbred matings in a totally random mating scenario would be even higher than this. So, and I should say that, so what the other thing that we did, and this is what this distribution is we said, well, okay, if you knew ahead of time what the pairwise coefficient of relatedness is between all individuals in that stock, and you could use that information to choose your mates, then what would the distribution be? And this is what the distribution would be. So this is your optimal distribution. Unachievable because of asynchrony in spawning and incomplete information and so on and so forth. But in principle, you could pretty dramatically shift the relatedness profile of your spawning pairs from here to there. So Morgan's already talked a little bit about genetic broodstock management, so I won't say too much about it, other than we derive these relatedness coefficients from the molecular genetic data, in this case 16 microsatellites, 18 in some cases, and then we rank each male, every single male, as a potential mate for every single female in order of how closely related they are. The least related males at the top and the more related males at the bottom. And you can see in some programs in some years, the proportion, the number of males that would create really highly inbred matings is very, very large. And these are the actual matings that occurred here for a year in 2004-2005 spawning season at the Russian River. And, oh, missed a little something there. And a few things didn't transfer over from my computer. And again, we can create these frequency histograms of the relatedness coefficients of randomly spawned individuals here in red, actually spawned individuals here in yellow, and the optimal spawn pairs if we had every male available to mate with every female throughout the spawning season. And so, as Morgan pointed out, we've done that now for 2 years at Iron Gate with the Coho stock and have substantially reduced the number of inbred matings. And in fact, Morgan already showed you this. I'll just show you the one for 2010. And again, no matings above the half-sibling level using this technique, and a dramatic reduction in inbred matings. Most of the matings are actually between individuals that are believed to be as unrelated as you can get in this stuff. So, in conclusion, the primary axis of population structure in the Klamath basin, this is upstream of the Trinity confluence because the populations in the lower basin and the Trinity River are doing their own thing, it is temporal and not geographic. And we believe that that is primarily a consequence of many years of restricting jacks for use as broodstock in the Iron Gate hatchery program. Turns out, and I was talking about the pedigree reconstruction project that we're doing, that there were some jacks that slipped through, not a whole lot, but some, and we can tell that through the genetic tagging pedigree reconstruction program. So they were probably, in their perception, doing what was part of their operational plan, were doing their best to not spawn 2-year-old fish. A few still got through and limited, although not very much, the temporal differentiation that has built up here. We also have demonstrated substantial gene flow between Iron Gate hatchery and natural populations of Coho salmon in the Klamath River. Like it or not, it's happened. And in particular, the Shasta River has been pretty strongly affected by Iron Gate fish. And one of the most important things that one can do in a situation like this is to try to minimize the fitness differential between hatchery fish and natural origin fish. That, honestly, if hatchery fish and natural origin fish had the same fitness in natural spawning areas, we wouldn't be that concerned about where hatchery fish go when they leave the hatchery. We'd be a lot less concerned, I should say. And so one of the most important things that we can do, given the situation that we have now, which is that we will never have 100% control of where hatchery fish go when they leave the hatchery. One of the very best things we can do is try to decrease the differential in fitness between hatchery fish and natural origin fish, so that when these hatchery origin fish spawn in natural areas, the loss of fitness that accrues to the natural population is as small as it can be. And this is a baby step in that direction, maybe more than the baby step. And we're happy to see it underway and having a measurable effect. So with that, I just need to thank a bunch of the people who have contributed to this, and a number, didn't have enough room to directly thank all the people who were involved in sample collection and funding and everything else, but this is a very large effort by a number of people, and they're all thanked for their contribution. I thank you all for your attention. Thanks.
Thank you very much. I suspect there may be a few questions for Carlos. Here's what I'd like to do, because we are going to have a panel discussion, and so some of the implications of his work for the broader questions you're all interested in, such as a suitable broodstock, we certainly can get into that there. What I'd like to do first is to allow the geneticists out there to talk shop with him, so that when we get to the panel discussion, you are all on as close to the same page as you can be, or at least you understand where you have differences of perspective. So, my geneticists, you've got Carlos, you've got his results. I know a lot of you have been waiting for them. Yes?
So, very impressed, Carlos. You've done a lot. That was really appreciated you running through.
You're doing fine, just
Thank you, really good job. Lots of data. One of the things if I compare what you have found and to what some of my colleagues in Oregon are finding, you're familiar with Michael Bluen's work. Yes. and Hitoshi's work, and how he's shown that selection and hatchery is a very strong effect. And
for steelhead.
Yeah, we have shown similar effects in Coho too, in the Umpqua. Mm-hmm. And some of those papers are coming out too. So it's apparent that the selection operating is really strong. So there's a bit of I guess I'm just being really open with you and sharing questions about what comes up for me, is that we sort of have two pulling forces, one selection for hatchery and one selection for whatever's going on in the wild. And so this integrated practice, bringing in fish that return to Iron Gate, where we don't know which local adaptation they might represent, is just sort of two selective forces that are strong and maybe counteracting. Is the problem that I'm wrestling with in my mind. I wanted to share it with you.
Those are all very valid points, very valid topic, and one that I think we're probably going to discuss a whole lot more in the panel. I should point out that I'm just reporting the patterns here and not making any value judgments about whether or not those patterns are favorable or detrimental or so on and so forth. As probably a number of you know, we're just finishing up a very comprehensive review of all of California's anadromous fish hatchery programs. Or, unfortunately, not all of them, but most of them, the ones in the Klamath Trinity and the Central Valley Basin. And one of the things that we struggle with a lot in that process, and one of the things that is sort of one of the defining features of the way that we treated our entire sort of analytical framework, was that it's just not possible to operate these hatchery programs and have them be entirely segregated from natural populations. We just are never 100% efficient at that. And the more that we try to separate them, the higher the fitness differential between the hatchery stock and the natural stock becomes, and the more detrimental introgression is for the natural stock when it's introgressed by a hatchery fish. That's not to say that we should have a free-for-all of hatchery fish breeding wherever we want, just that the concept of a completely segregated hatchery program is not really achievable in California, and that it's something that we need to get past as an ideal, because it just doesn't work. Even if we set up barrier weirs and we have folks out there doing marked selected fisheries or whatever the case is, you always will have some fish that are going to go over the weirs when it overtops, or are going to escape harvest, and you just can't segregate them entirely. So we've got this issue, like it or not, that we have to deal with. And the question becomes, what's the best way to deal with it? And that's kind of an open question. My comment at the end was that one thing that we know we can do is try to reduce that fitness differential, and one simple way that we can do that using molecular genetic data is simply to reduce the rate of inbreeding in the hatchery and the consequent inbreeding depression and fitness loss that is associated with it. And that's, as I mentioned, that's a baby step, maybe a little bit more than a baby, a toddler step, but it's a step in the right direction, certainly not all the way there.
I also have a question in the back.
I'm Sean Durham, a geneticist from the Columbia River Basin. Carlos, I think you did an excellent job. This is a really thorough analysis, not only with the number of samples in this small geographic region, but also the extensive number of markers that have been used to evaluate the population structure and gene flow. I was curious if you looked at any trends of genetic diversity as far as measures of heterozygosity and allelic richness. And based on those patterns, I wouldn't expect there to be major differences in heterozygosity, but I'm particularly curious about Iron Gate hatchery, if you see any kind of evidence for lower diversity in those hatchery brood years.
So the answer is that we haven't, well, we have looked at just simple measures of allelic diversity. There is a little bit less in recent years in the 2006, 2009, for which we only have 2 samples, probably because of the much smaller number of fish that were returned and spawned that year, so it's not surprising. But by and large, there are confounding issues, and one of the confounding issues is the natural origin fish, and we don't necessarily know exactly what we now have a little bit of information, but we have not yet, we didn't have the information available to us about the proportion of natural origin fish and which ones were natural origin fish, as well as the fish that came from the Trinity River and were integrated in a broodstock, of which you might have seen there were a fair number. Well, not that many, but there were some, they were sprinkled throughout the brood year. And really, without knowing which fish those are and sort of excluding them from your calculations, you're going to get kind of an inaccurate picture. So we haven't paid that much attention to it at this point. This is really more of a structure analysis. And as you know, there are so many pitfalls involved in trying to estimate relative levels of genetic diversity. Like for the juvenile samples, we know there are siblings in those sets of juvenile samples. We have not gone through the analysis yet to weed those out and to then try to calculate allelic diversity on the basis of just what we believe are the unrelated individuals, which is what you would need to do to get at that question. So the answer is there's a little bit of a signal of a reduced genetic variation in one of those brood cycles, but by and large, they seem to be fairly similar, and we don't really know whether there are any fine-scale differences. So.
I guess, a similar type of thing, any evidence for that in the natural, or I guess any of the spawning aggregates from Shasta or Scott Creek or any of the other collections?
Well, Scott Creek, you mean the Scott River? Yeah, Scott River. Well, we don't have temporal samples there for the most part, other than just a very small handful of samples from a couple of years, and not from the same tributaries. So we can't really do a temporal evaluation there. And again, without going through the process of identifying siblings, and we've actually developed an algorithm for doing that quite recently, and we've got a paper that we're just submitting to show how effective it is. Going through, figuring out what the sibling groups are, correcting for that, and then using those unrelated individuals as the basis for estimating the relative genetic diversity. We can't really say at this point. Is the lower, for example, yeah, there is significantly lower genetic variation in the sample from Beaver Creek, but is that simply due to the fact that we sampled one or a couple of very large sib ships? We found sib ships of more than 25 individuals in some samples from other basins. And if it is, then it might not have anything to do with the relative genetic diversity of the subpopulation or the population per se, but just be a family effect. So without correcting for that, which again we have not done yet, we can't really say.
So in that example, was that population or that collection out of Hardy-Weinberg equilibrium when you
Yes.
have a lot of, yes?
Yes.
Okay.
But
Yeah.
pretty much all of the juvenile collections of salmonids in California that we encounter are out of Harding-Weinberg until we do this SIP correction.
Sure.
So.
Question back here.
Mhm.
I'm Christian Smith. I had one sort of clarification. So the structure, I mean, it was pretty obvious that it was predominantly temporal rather than geographic. When you looked, or did you look at pairwise tests of heterogeneity or allele frequency significance or FST significance to ask whether what geographic structure was there was significant?
Everything is significant except for the parent-offspring brood cycle comparisons at Iron Gate.
Thanks.
Yeah.
All right, last call for geneticists. Okay. Other scientists with a question.
Carlos, could you clarify what you just said when you said everything is significant?
Oh,
I'm sorry.
So the FST values, the measure of pairwise differentiation is significantly different from 0 in every case. So you can have a positive value, but it's so small that it's actually, and it's, when you actually test to see whether it's significantly different than 0, it may or may not be. And that was his question. So you can have positive values, including ones that can range up to almost 1% differentiation that are not statistically different from 0. Those are all different, except for that.
Can I ask one more question? It was either Christian or Sean referred to the temporal versus the spatial structuring. Just, seeing the charts, I guess, I didn't know how to interpret that because it looked like the only brood years you had up there were related to Iron Gate.
Oh yeah,
Right.
that's why at the end I showed you, popped those little red brood cycle, brood year assignments onto the natural population, so that you
okay.
could see that the natural populations were all clustering with the Iron Gate brood year, or brood cycles that they, with which they shared, with the Iron Gate fish with which they shared a brood cycle.
Right,
And-
right.
But perhaps more importantly, that the other thing to note is that the fact that you don't have the Iron Gate brood cycles clustering together and the natural populations clustering together is perhaps a strong evidence that it's evidence of two things. One, that the temporal variation is the largest axis of variation in the Klamath, and also that Iron Gate fish have introgressed natural populations in the upper Klamath Basin quite substantially. And that's a qualitative term, intentional. So.
Well, I was going to ask you about that too. Go ahead, Rich.
Did you do any examination in, I think it would be the FST graphics, whether there was any spatial pattern for the Iron Gate fish being fish like from the Scott River being classified as Iron Gate in terms of distances or how many turns they had to make to get to a wrong place or anything like that?
Not in terms of terms, but Bogus Creek is essentially a satellite population of Iron Gate. And aside from that, the Shasta River is the one that, if you remember these, these here, the Shasta. Well, let's go to the. If you look here, that's where you see it most. It's easiest to see. I'm sorry, the Shastas. These were done alphabetically instead of by geography. So the Shasta is stuck right in between a bunch of Scott River populations, but you can see clearly that it's been quite introgressed from iron gate fish. And again, yeah, the temporal axis of variation is very apparent there. So yeah, the answer to your question is Bogus and Shasta, which are geographically the closest as well.
So so I understand the statistical differences. When you say quite integress, you could use the term quite integress, but it would still have a statistically significant FST difference.
Yeah, oh yes.
Okay.
Absolutely.
Okay.
Yes.
I'm John Menke. I'm a retired professor at UC Davis. It's been about 20 years we've had low brood year and high brood year runs, too low and too high, like on Scott. I think they also show up on Shasta until the population got so low, and also returns to Iron Gate. Do you see differential degrees of inbreeding for the lower brood years compared to the high brood year sizes, indicating that there are less mate choices and are we starting to see a decline or increase inbreeding on the brood years that have very low numbers of fish?
That's a really good question. We haven't actually really looked at that yet. And again, there's this confounding issue that we literally have just been working with Morgan Knockley, and he's been working, I guess, with Mark Hampton, who had his position previously, to determine which of the fish in the samples that we have analyzed from Iron Gate are natural origin and which of them come from other stocks, some of which were spawned previously. Because, again, without accounting for those, it's really hard to make that determination. Because those fish are going to be, sort of by definition, a lot less related to the Iron Gate produced fish than the Iron Gate produced fish are to each other. So that's an important question. I will say, and I think I pulled that out of my talk for tomorrow, but we have at Warm Springs, we've been able to measure this with the Coho salmon stock there, and we saw that we could measure how diversity was decreasing until we started genetic broodstock management, and then we sort of arrested it. We didn't, as Michael Lacey pointed out, we don't have a mechanism, or we do now actually, and we'll talk a little bit about that, but we didn't have a mechanism to actually increase it without bringing in fish from somewhere else, but we were able to arrest it.
Okay, 2 more questions, then we're going to take our break, and then we're going to move on to the panel discussion. So you had a question?
Has a similar analysis been done on like the Columbia, and or are these the FST values you see on the Klamath comparable to what you see in these other big watersheds?
Well, the Columbia is kind of a funny example because they lost most of their Coho salmon and they've now rebounded. Actually, Sean Narum in the back, and I think also Christian, work on Columbia River Coho, and they'd be much better positioned to answer that question than I. Although I will note that in places like the Eel River and other smaller basins, we see similar levels of gene flow between tributaries. That really, in Coho salmon, and this is a little bit of a deviation, and this is why the Iron Gate, the Upper Klamath example is so striking. By and large, we see a very strict hierarchical structure in Coho salmon of between basins, tributaries within basins, brood cycles, and then brood years. Is that right? No, brood years, then brood cycles.
These are all your samples come from adults, right?
No, a lot of the natural, in fact, almost all of the natural population samples are from juveniles. No, they're with the exception of the Scott and the Shasta, where at least some of them are carcasses.
Because it seems like the data could be a reflection, like if you have an increased rate of gene flow in, say, the Shasta, and you're sampling adults, well, that could be stem from the fact that there's a high rate of juvenile mortality. So there's no native fish coming back because y'all got whacked with smolts, and all there is to sample are strays from Iron Gate or from other places.
If you're sampling adults, you're talking about.
Yeah.
Yeah, that's
But that's not, you're saying that's not really reflected in this data set because overwhelmingly sampled juveniles.
Well, the Scott River samples and the Shasta River samples, I think actually maybe all of the Shasta River samples with the exception of like 5. I mean, it's undersampled. I think we have 60 or 70 samples as opposed to 300 from the Scott. Almost all of those are juvenile samples in the Shasta River, so they were produced there for the most part. And some fraction of them were undoubtedly produced by strays from Iron Gate. So.
I thought you had gotten some more recent samples from the Shasta that perhaps were not from the canyon reach, but from more up at the springs.
From Big Springs, yes, that's right.
Right.
Those were included there, yes.
So did you see any, did you check for
Same
heterogeneity
thing.
between them?
Those are just would be the second half of that group there.
So they basically lump in with Shasta, Iron Gate fish?
That's right, yeah.
Yeah.
That is, yeah. That's these fish. Yeah.
So it shows no difference between Canyon and Big Springs?
Yeah, the
As far
first
as
set of, yeah, pretty much, yeah.
And then I had one more question if I could. So you looked at Iron Gate, Shasta, Bogus, which they're all pretty close.
Yeah,
probably
sorry
no-
if you can't see these, I wasn't able to actually.
Right.
Bogus, Beaver, Blue, these are all Iron Gate, McGarvey, Noy Orion, Scott River with the Shasta stuck right in the middle, and then the 2 Trinities at the end. So it's the way that this program spits it out.
So the beaver sample size was adequate, you feel, or?
Yeah, you know, more is always better from a geneticist perspective. It's not an outstanding sample size, but they do cluster very strongly with the Iron Gate brood cycle with which same year that they were sampled.
Okay, well, let's thank Carlos again, please.